Hi everyone, I hope you all are doing well. In this lecture, we're going to complete chapter 4. This is chapter 4, part 2 of the chapter Psychopharmacology. Now, bear with me through this lecture. This might be the most challenging chapter material of our entire semester. And I say that because there's just a lot of terms. There's a lot of terms. In this lecture, we're going to learn about neurotransmitters. We're also going to learn about their receptor sites in the body, so the different sites that actually receive the neurotransmitters. We're going to learn about what are called precursors to those neurotransmitters, so what comes together to form the neurotransmitters, and then also what then breaks down the neurotransmitter and removes it from the synapse. We're also going to learn about a variety of different drugs and how they interact with neurotransmitters, as well as how they interact with their receptor sites and how they may also interact with the removal of the neurotransmitter away from the synapse. So there's a lot of stuff in this particular portion of lecture, but just bear in mind, as always, this is just to introduce you to these concepts and ideas. Please don't get too bogged down into the terms, and just try to get the general gist of what we're talking about today. And feel free to watch this lecture more than once if you need to, go through your chapter material more than you need to, or more than you normally would if you really feel as though this is pretty sticky. But just keep in mind that my expectations overall are fairly modest, as I know that most all of this is going to be very, very new to everybody. And so let's just learn about neurotransmitters, do the best we can, and have a good time. First, let's start with what is a neurotransmitter. A neurotransmitter is a chemical that is released by the terminal buttons of a neuron into the synapse, or the gap between two neurons. Now, up until very recently, most researchers thought that neurons only released or received a single neurotransmitter. For example, a neurotransmitter that we'll talk about today is dopamine. So most researchers believed that if a neuron released dopamine or received dopamine, that it could only receive or release dopamine. But what we know now, very recently, is that it might not be quite that simple. For example, a cell that releases dopamine might also release something else that's accompanying that. Now, I'm not going to dig too deeply into that today in our lecture, but just know that it may not be as simple as a single neurotransmitter for a single cell. But for our learning of what the different neurotransmitters are and what the receptor sites for those neurotransmitters are, we will take kind of more of that focus just so things are clear. But just know that the story may not be quite that simple. And I will add one more thing, is that we are discovering things all the time. They're still actually discovering neurotransmitters to this day. So this is an ever-evolving field of study. Now we'll take a look quickly at how drugs can interfere with the process of neurotransmitter production and transmission across neurons. Precursor drugs are drugs that are meant to increase the amount of neurotransmitter that any given cell can receive and break down, and then release back into that synapse, or that gap between neurons. These precursor drugs can be either an agonist or an antagonist, and I mentioned that at the end of our lecture last week. But just as a reminder, if a drug is an agonist, it's going to increase activity or be more excitatory, whereas if it's antagonistic, it's going to inhibit a process or stop things, slow things down. So let's zoom in on two neurons. Here we have the sending neuron and the receiving neuron. Or you could also think about that as the postsynaptic neuron. So this is the neuron that's going to be receiving the neurotransmitter, and then this is the sending one, or the presynaptic neuron that is sending the neurotransmitter. Now, during action potential, neurotransmitters that are stored in these little round things here called vesicles, these will bind with the presynaptic membrane here. So this membrane of the sending cell, and open up and release through these transporter sites. They will open and release the neurotransmitter into the synapse here, the gap between the neurons. And then from there, if things are working as they should, there are receptor sites on the receiving neuron. And the neurotransmitter will bind to these receptor sites and then activate activity in the receiving neuron. Now, as I mentioned a couple of slides ago, there are drugs that interfere with this basic process of how a neurotransmitter is released into the synapse and then taken in by the receiving neuron. For example, some drugs block the vesicle transporters by binding and basically preventing the vesicles from refilling back with neurotransmitter. So these are antagonistic. So these are going to kind of block activity. So what happens with the use of drugs like this is that these vesicles that I mentioned in the previous slide that are filled with the neurotransmitter through reuptake, or basically what happens when the neurotransmitter has been released into this synapse, with reuptake, it's taken back in by the sending cell, if you all will remember from chapters before. Now that's the normal process, but if we have a drug that it's antagonistic and it's going to bind with the transporter sites here, so for example, we have this inhibiting drug, it binds with the transporter site, so what this does is it doesn't allow the neurotransmitter to be reuptaken back into the sending cell. So then what happens is that these vesicles don't ever refill with the neurotransmitter. So then during action potential in the future, nothing happens. So the neurotransmitter can't be released back out. Now, some drugs prevent the release of the neurotransmitter from the terminal button altogether. These are also antagonistic. Remember, they are impeding the process or inhibiting process. So what these do is deactivate the proteins that allow for cells to basically dock together. So they deactivate these proteins and basically the neurons don't fuse together. So there's no fusion between the presynaptic and postsynaptic membrane, and so then there is no release then of any neurotransmitter into the synaptic cleft. So release of neurotransmitter just doesn't happen. Let's take a look at the effect on receptor sites. So here we're looking at the receiving neuron or the postsynaptic neuron. As I mentioned, there are receptor sites on the receiving neuron that are there waiting to receive the neurotransmitter. Now, some drugs can be basically competitive with a neurotransmitter. So for example, we can have a drug that is a direct agonist. So what this is going to do is be excitatory. So if it is a direct agonist, what it's going to do is going to bind on the receptor site right where the actual neurotransmitter would bind. So instead of the neurotransmitter binding on this receptor site, the drug binds to the receptor site. And in this case, it's going to mimic the effect of the neurotransmitter. So it's going to, again, be excitatory and basically activate the neuron just as the neurotransmitter would. For example, nicotine mimics the effect of something called acetylcholine. Acetylcholine is a type of neurotransmitter, which we'll discuss in a few minutes. But for now, just know that the drug nicotine mimics the effect of the neurotransmitter acetylcholine. And it does that by binding to the site where acetylcholine would bind. We can also have a drug be a direct antagonist. So in this case, the drug will again bind where the neurotransmitter would typically bind. But instead of being an excitatory response or reaction, instead we're going to get inhibition. So again, this is going to be antagonistic. So it's going to bind where the neurotransmitter would, but the result of that is going to be an inhibitory one. Here we'll look at a couple of more different ways in which we can have an effect on receptors by drugs. So again, we're looking at the postsynaptic receptors or the receiving cell. We can also have what is called non-competitive binding. The two examples that we looked at previously on the last slide are referred to as being competitive binding because the drug binds exactly where the neurotransmitter would bind. In the two cases that we're looking at here on the right-hand side of your slide, these are called non-competitive binding because they don't take the place of the binding site of the neurotransmitter, these drugs. So if we'll take a look over here, in this example, the neurotransmitter binding site's right here, but the drug doesn't bind here. It binds over here. Same thing for this example here. Here's the neurotransmitter binding site, but the drug binds on the other side. We can have an instance where this is going to be antagonistic. We could also have instances where it might be agonistic. So it might be an inhibitory or an excitatory response. So if it is an indirect antagonistic response, now we say indirect because it is not binding to the neurotransmitter receiving site here. So an indirect. And if it's an indirect antagonist, what that's going to do is, again, bind to a site that is not the neurotransmitter site, but it is going to inhibit the effects of the neurotransmitter. It's going to produce an inhibitory response. So, for example, another neurotransmitter that we're going to talk about here in a few minutes is glutamate. For glutamate, there are indirect antagonists called PCP and ketamine. You may have heard of these before. These are drugs that actually prevent the effects of glutamate. And they do this by indirectly binding to a receiving site on a receptor. The flip would be as if we have something that is also indirect but produces an excitatory response. So we again say indirect because the drug is not binding to the neurotransmitter receiving site. It's binding somewhere else. But in this case, we're getting an excitatory response. An example of this would be the drug called diazepam. This happens concurrently on neurons that receive what is called GABA. GABA is another neurotransmitter that we will talk about here in a few minutes. GABA typically has an inhibitory reaction in the body. Basically, if you want to think about it more as being relaxing. Diazepam increases GABA's efficiency. So it mimics the effects of GABA and produces more of the effects that GABA produces. So because we have an excitatory response or an increase in behavior, diazepam is an agonist. And again, it's indirect because it's not binding to the neurotransmitter receiving site. It's binding somewhere else on the receptor. We can also have instances where some drugs prevent reuptake, as I mentioned earlier in this lecture. Some drugs prevent reuptake of the neurotransmitter back into the sending cell or the presynaptic cell, which here is this one right here on the top. A good example of this is cocaine. So what cocaine does is basically it binds with the enzymes that destroy a neurotransmitter. So if we're looking at the synapse here, cocaine is represented by these little red kind of building block looking things. And then we have dopamine. Dopamine is another neurotransmitter that we'll discuss in this lecture. So dopamine, again, is a neurotransmitter, so it is being released into the synapse and it's hanging out here. But then we also have cocaine in the system, and what cocaine is doing is it's basically binding with the transporter sites here, and it's blocking reuptake of dopamine. So dopamine can't be taken back in. The result here is a lot of neurotransmitter, again, dopamine in this case, hanging out in the synaptic cleft. This isn't a good thing. This could actually cause a lot of hyperactivity and other issues because dopamine's not being cleared out. So what happens is that because it's hanging out longer, it could potentially be taken in more by surrounding neurons. Now let's chat about some of the different neurotransmitters that we have in our body. Firstly, there are many of them, but most synaptic communication, so neuron to neuron communication, is done by glutamate, which is an excitatory neurotransmitter. This is a chemical that, when released, produces more activity or excitation. Also, GABA, or gamma aminobutyric acid, we'll call it GABA for short. This is an inhibitory neurotransmitter. So this is more of that calming chemical. This is going to reduce activity. And then finally, glycine. This is also inhibitory, and it's found in the spinal cord and lower brainstem. Now, throughout activity, there really strives to be a balance between excitation and inhibition. That is, your body's trying to stay somewhat in balance. We don't want too much excitation, but we also don't want too much inhibition either. So your body is basically trying to find a balance between these two processes of excitation and inhibition. All of your sense organs, or your PNS, basically, your peripheral nervous system, all of your sense organs transmit information to your brain, or signals to your brain via synaptic communication, so neurons sending messages or signals from neuron to neuron. They transmit this information to your brain through axons that release glutamate. With one exception, those that receive painful stimuli. This is done through a different protein. But what this tells us is that the glutamate system, that is all of the neurons that send and release glutamate, there's a lot of them because all of your sense organs transmit to the brain through axons that release glutamate. So glutamate is probably the most prevalent neurotransmitter, followed quickly by GABA, which is that inhibitory neurotransmitter. Now, what we know is that most cells are organized into specific pathways. Specifically, there are neurotransmitter pathways. So, for example, there are glutamate pathways, there are GABA pathways, and we can learn through drug studies what those pathways look like, where they project to, because what we do know is that most cells are organized in a pathway such that they originate in one or more brain regions, but then they project into other regions. So we can actually learn how these pathways are organized within our body using drug studies, and I'll mention those in a little bit later in lecture. So this is a figure out of your text. I wanted to put it up in lecture as well, just to encourage you to take a look at it. This is listing out some neurotransmitters, many of which we will discuss here basically right after the slide. But I wanted to show you this and kind of encourage you to take a look. It's listing out the neurotransmitters, but also some of the examples of how they function in your central nervous system, as well as how they function in your peripheral nervous system. So kind of what role do they play? What do they do? We'll talk about that now moving forward, but this is a nice condensed version of all of that information that's very quick to digest. So I encourage you to take a look. Now, let's chat about some neurotransmitters more in depth. So let's focus on the first grouping of neurotransmitters, which are amino acids. Amino acids, these are used by all of your brain cells for protein synthesis or protein breakdown. We suspect that there's about 8 that might serve as neurotransmitters for mammalian central nervous systems like ours. Most common are the ones I've already mentioned, glutamate and GABA. Okay, let's first start with the neurotransmitter that I've been referencing the most throughout this lecture thus far, glutamate. So as I've mentioned, glutamate is going to produce an excitatory reaction in the body. We find that glutamate is largely allocated to neurons in the brain and in the spinal cord, so in the central nervous system. And again, this is going to yield more firing in the neurons in this area, so an excitatory or active response in the body to glutamate. Glutamate is synthesized from a precursor called glutamine, and it is done so by the enzyme glutaminase. So glutaminase is the enzyme that takes glutamine and then creates glutamate. There are 4 major types of receptors in your body that bind with glutamate. 3 of them are what we refer to as being ionotropic. What that means is that when glutamate binds with these receptor sites, the ion channels open and allow for action potential to happen. So we also call this direct binding because as soon as glutamate binds to these sites, the ion channels will open. These 3 are NMDA, and your text talks about NMDA the most, AMPA, and kainate. There is an additional 4th receptor called metabotropic glutamate receptor. So this particular receptor is different than the previous 3 in that there's an additional step involved for action potential to happen or for those ion channels to open. So for this type of receptor, when glutamate binds to it, it actually doesn't immediately open the ion channels. Instead, what happens is that a chemical is produced, and that chemical then opens the ion channels and allows for activity or action potential to happen. Now, glutamate is removed from the synapse or the gap between neurons by acid transporters and then broken back down into its precursor, glutamine. Now, if there is a failure to remove glutamate, so if glutamate is allowed to hang out in the synapse, as you can imagine, it's an excitatory neurotransmitter. It's going to yield more action potentials, more firing in neurons. So we might wind up with what looks to be more hyperactivity. This can be referred to as glutamate exotoxicity. So basically what happens is that the neurons are receiving too much glutamate. So they're firing way more than they should, and this can lead to overexcitation and damage to these neurons. We think that this might be what plays a role in brain damage as a function of stroke, and also in ALS. ALS is more commonly referred to as Lou Gehrig's disease. So with this particular disease, there's damage in the neurons in the brain and in the spinal cord. And we think, again, it's happening because of glutamate exotoxicity. And what happens here is a loss of muscle control over time. The drug given for this, if you're interested in this sort of thing, as your text discussed throughout, different drugs that are given to address these issues. One drug that is given to ALS patients is called Rilotec. And what happens here is with Rilotec, the goal of this drug is to reduce the amount of glutamate that actually gets to the sending neurons membrane. So the little vesicles that we took a look at earlier that hold the neurotransmitter, those vesicles that are holding glutamate, if you're taking Rilotec, they don't ever actually get to the presynaptic membrane or the sending cell's membrane. And because they don't ever get to that membrane, they can't be released into the synaptic cleft. Now let's take a look at the other neurotransmitter that I mentioned earlier, GABA. GABA is, in contrast to glutamate, inhibitory. So this is going to be more of that calming reaction in the body. So this is very widespread in your CNS through your brain and your spinal cord. It is produced from a glutamic acid by the enzyme GAD for short, or glutamic acid decarboxylase. So what is happening here is you have the enzyme GAD that is taking the precursor glutamic acid and synthesizing it and making it into GABA. For an example of a drug that interacts with the GABA system, and this is more of an exploratory drug, we tend not to prescribe this to patients. It's called alliglycine. What it does is prevent the synthesis of GABA. So it prevents the act of the enzyme GAD from producing GABA from glutamic acid. So GABA is never synthesized. It's never actually made. And what this does, again, we don't really prescribe this to patients for anything. We want to understand these systems more. So this is more of an exploratory drug, and it's meant to help us understand how the GABA system works in the body. We, again, use it very little for patients because what it is likely to do is increase the likelihood of seizures. Remember, GABA produces an inhibitory effect. So if you think about muscle movements, it's going to depress those or slow those. So if you give a drug that prevents GABA, then you're not going to have that inhibitory reaction through the body. So you're going to have more muscle movement, more activity. So for someone who already suffers seizures, it could actually increase the likelihood that they would have even more seizures. Now, GABA secreting neurons, as you may have already guessed thus far, inhibit excitatory neurons. So basically the signal that GABA is giving is don't fire, calm down, don't fire. Here we're taking a look at a picture from your textbook of a GABA receptor. A GABA receptor here, as we're taking a look at it, if you'll notice, there's more than one receptor site. The main site is for GABA itself, but there are also other sites that will affect this GABA receptor as well. If you remember when we discussed benzodiazepines and barbiturates in a previous lecture, these are classes of drugs that have been given in the past or even more recently, benzodiazepines for anti-anxiety medication. These drugs interact with the GABA receptor and actually promote GABA or increase the availability of GABA and reception of GABA. So GABA, again, is going to produce an inhibitory reaction from the body or a calming reaction from the body. It's going to calm down the excitatory neurons or the excitatory system and produce a calming effect. That makes sense for an anti-anxiety medication. Keep in mind that barbiturates aren't really used as much anymore because they have a very low therapeutic index, as we discussed previously. And again, just as a refresher, what that means is that with a low therapeutic index, it's much more likely that you could have negative effects or toxic effects, such as overdose. So barbiturates are more dangerous. Benzodiazepines are used more in present day for anxiety medication. And again, they work by promoting GABA, by binding to the GABA receptor. And you can see the site for benzodiazepine and barbiturate here. Related to benzodiazepines are the drugs commonly given to increase sleep or help with sleep, Ambien and Lunesta. These aren't exactly the same, but they work similarly to benzodiazepines in that they promote GABA. Now on the flip, you see a site for what's called picrotoxin. This inhibits GABA. So this doesn't promote GABA, it inhibits GABA or inhibits that calming effect through the body. This can cause seizures potentially, as you can imagine, because that inhibitory effect on the excitatory neurons isn't happening because GABA is being inhibited with this particular drug. There is also a site here on this GABA receptor for a steroid hormone. And when binding here happens, we get a relaxing anxiolytic effect. So this is going to promote GABA. We see increase of this in women in pregnancy. So we see an increase in the steroid hormone binding, and that makes sense because it's going to produce more of a relaxing effect. And we could see how something like that may have evolved and may be beneficial for a pregnant woman. Now, when GABA needs to be removed, it is removed from the synapse and then broken down by what is called GABA aminotransferase. So that's the enzyme that's going to break down GABA and get rid of it from the synapse. A couple of drugs are given to interact with this process, this removal of GABA. The first is Gabatril, which is the name brand. This is a transporter antagonist, meaning that it is going to prevent the transportation of GABA from the synapse. So it's going to allow GABA to hang out in the synapse. This is going to increase GABA availability because it's not being removed out by, this drug is being used to prevent the removal of GABA. So then GABA is allowed to hang out in the synapse. It increases GABA availability, and this is given commonly to reduce seizures. So the more GABA that's allowed to hang out, the more likelihood that GABA is going to be able to bind with the neighboring neurons and produce that inhibitory effect of the excitatory neurons, that relaxing effect. Another drug blocks GABA aminotransferase, so it's going to produce the same type of effect. Gabatril inhibits the transportation of GABA out of the synapse, whereas Sabril actually blocks the enzyme that breaks down GABA. So the result is going to be the same. More GABA is going to be allowed to be available. This drug is also given for seizures as well as commonly given to epileptics as well. Now we move on to another neurotransmitter called acetylcholine. Acetylcholine is found in very specific pathways in the CNS. Acetylcholine typically has what we call more facilitatory effects. So they tend to facilitate behaviors that we engage in. So REM sleep, for example, we find a acetylcholine pathway in the dorsolateral pons in the brain. We think that this is helping to facilitate REM sleep. We also find another pathway in the basal forebrain. We think that this is meant to help with a facilitation of learning. And then we also find another pathway in the medial septum. We think that this is helpful for memory formation. So this is helping to control the electrical rhythms of the hippocampus. So if you notice, acetylcholine seems to be a bit more specific and have more facilitatory effects for behaviors that we engage in. Now, the primary neurotransmitter, acetylcholine here, is secreted by axons of the peripheral nervous system to stop muscle contraction. Now that makes sense if we're thinking about REM sleep, for example. If this neurotransmitter is secreted by axons in the peripheral nervous system and is meant to stop muscle contraction, well, that makes sense for REM sleep, that would help facilitate REM sleep because it is slowing muscle contraction. The precursors to acetylcholine are choline and acetyl coenzyme A. These two things come together to form acetylcholine. That is done so by choline acetyltransferase, or CHAT for short. This is what produces acetylcholine from choline and acetyl coenzyme A. Now, interestingly, we've been talking about drug interactions with neurotransmitters and receptor sites throughout this lecture thus far. What's interesting about acetylcholine is that there hasn't really been any significant drug developments to interact with acetylcholine, or specifically, I'm sorry, not acetylcholine, but choline acetyltransferase. So the enzyme that actually produces acetylcholine. There hasn't really been any drug development to interact with that. But what we do see used is something called Botox, or botulinum toxin. This is actually a toxin that has been diluted quite a bit and is injected in order to prevent the release of acetylcholine. What this will do is cause muscle paralysis, and this is of course used for cosmetic reasons, also used for migraine treatments. But what this can do in large doses is called suffocation and paralysis, because it is going to prevent acetylcholine from being released. So this is going to increase the likelihood of muscles not being able to move the way that they need to and actually cause paralysis. A lesser version is black widow venom. This is actually less toxic than Botox is on its own. It actually does the opposite. It affects release in that it stimulates release of acetylcholine. So whereas Botox prevents the release of acetylcholine, black widow venom stimulates the release of acetylcholine. So rather than cause paralysis, it's going to cause more muscle movement. And again, this is less toxic than botulism toxin or Botox is. There are two different types of receptors that receive acetylcholine. One is inotropic and one is metabotropic. And if you remember from previously in this lecture, inotropic means that the acetylcholine binds to the receptor site and ion channels open and action potential happens. So it's a direct binding. Indirect binding with metabotropic is going to create that additional step. So if acetylcholine binds to the receptor site, that doesn't open the ion channels directly. Instead, it produces a substance, and then that substance will open the ion channels and allow for action potential to happen. So one is more immediate, inotropic, and one takes a little bit longer to happen, so metabotropic. Now, the inotropic is actually stimulated by nicotine. And we see this much more in your peripheral nervous system. If you think about it, muscles in your peripheral nervous system need to be very reactive, need to be able to react quickly. The inotropic receptor sites are going to act more quickly because there's not that second step involved. So there's more inotropic receptor sites in your peripheral nervous system, and they are stimulated by nicotine. The metabotropic ones are more so in your central nervous system. They are stimulated by what is called muscarine, which is a drug found in a poisonous mushroom, actually. So they are called muscarinic receptors. And again, this is more in your CNS. You do have some inotropic receptors in your CNS, but again, you have more muscarine receptors there. So again, your peripheral nervous system is going to contain more rapid acting receptors, so that inotropic receptors that respond more to nicotine. And again, your CNS contains both kinds, but more muscarine. Now, there are two classes of drugs that can interact with your acetylcholine receptors, specifically blockers. So the drug atropine blocks the muscarine receptors. One effect of this is to reduce saliva production. This is given sometimes in surgery. And then there is another drug called Curar, which blocks nicotine receptors. This can actually cause paralysis. So we want to be careful with Curar. But the effects of Curar are much faster than botulism toxin, for example. Now, lastly, how do we get rid of acetylcholine from the synapse? It is reuptaken by acetylcholinesterase. So this is going to be what allows for the reuptake of acetylcholine. There is a drug that can deactivate acetylcholinesterase. It is called neostigmine. This inhibits the acetylcholinesterase, so the enzyme that's going to take away acetylcholine. It actually inhibits that enzyme. And this drug is given to treat what is called myasthenia gravis. So this is a very severe condition where the immune system actually attacks your acetylcholine receptors in your skeletal muscles. So this particular drug is meant to inhibit the attack on the acetylcholine receptors. And it does that by interacting with or deactivating the acetylcholinesterase that gets rid of acetylcholine in your system. Now we move on to a different type of a class of neurotransmitters, if you want to think about it like that. These are called monoamines. The ones that we've been discussing before are amino acids. These are called monoamines. These are produced by several neuron systems in the brain. They mostly consist of smaller cell bodies in the brainstem, but their axons project out to different areas of the brain, and they give rise to a lot of different terminal buttons. The different monoamines are listed here. You've probably heard of all of them. We have dopamine, norepinephrine, epinephrine, serotonin, and histamine. The top three are a subgroup of monoamine called catecholamine. First, let's take a look at dopamine. From what you have probably learned about dopamine thus far, you may assume that dopamine is going to produce an excitatory effect. However, dopamine can be an excitatory neurotransmitter most of the time, but occasionally it can be inhibitory. It's going to depend upon what cell is receiving it. So it's going to depend on the postsynaptic receptor. whether or not it produces an excitatory or an inhibitory effect. We think that dopamine is heavily involved in things like movement, the learning or acquisition of new tasks, attention, and drug reinforcement. There are a couple of important pathways through the brain that dopamine takes. The first is through the substantia nigra. Through this area, it can project into the basal ganglia, which is heavily involved in movement, and then it can also project into the caudate nucleus and the putamen. The other pathway goes through the ventral tegmental area, or projects into this area. From here, we have two different systems that are located in this particular area. One is more related to the reward pathway that's going to involve the amygdala. And if you remember from previous lecture, the amygdala is heavily involved in emotions and emotional regulation. So this makes sense with the reward pathway. The other one is more involved in short-term memory and problem solving and planning. Now here we take a look at the precursors to dopamine, so how dopamine is actually formed. The precursor molecule is slightly modified step by step in order to create dopamine. So first we start with tyrosine, and you can see that on the top of our figure here that we're looking at on the right-hand side of the screen. So tyrosine is modified by tyrosine hydroxylase to become L-DOPA. You may have heard of L-DOPA before. L-DOPA is then modified by dopa decarboxylase to become dopamine. Now from there, dopamine can be converted into norepinephrine, and it can also then further, norepinephrine can be interacted with and made into epinephrine. There are drugs that interact with this whole system. For example, the drug AMPT basically inactivates tyrosine hydroxylase. And we can see along the timeline here on the right, that's going to deactivate this right here. So if tyrosine hydroxylase is deactivated, then what that means is that tyrosine can't be modified into L-DOPA and then later be modified into dopamine, and then further on, if necessary, into norepinephrine and epinephrine. So it completely breaks down this whole system. And then also another drug, L-DOPA, is used commonly for Parkinson's patients. The interesting thing about L-DOPA is that it can cross the blood-brain barrier. If you remember the blood-brain barrier from earlier lecture, the gaps in the capillaries there are basically non-existent. Not much can pass through the capillaries in your brain or your blood-brain barrier. L-DOPA can, but dopamine can't. So L-DOPA is given for Parkinson's patients because it can cross that blood-brain barrier. From there, it can be taken up by dopaminergic neurons or neurons that receive dopamine. And then it can be converted into dopamine that way. So L-DOPA is given, and it's allowed to cross into the brain or cross through the blood-brain barrier. And then by dopamine receiving neurons, it can be received and converted into dopamine, which can help with Parkinson's symptoms like tremor and things like that. So I'm showing you this very complicated process so you can see why dopamine, norepinephrine, and epinephrine were grouped into that separate grouping called catecholamines. And that is because you can see here that it's kind of a step-by-step system. So one becomes what it becomes because of the pre-existing step. So norepinephrine comes from dopamine. Epinephrine comes from norepinephrine. All of these are kind of a step-by-step process and are linked together. Now, in terms of dopamine receptors, we have identified 5, and they're just called D1 through D5. D1 and 2 are the most common. D1 and 5, we think stimulate or increase production of second messenger. So that substance that's formed after binding that's going to help the ion channels open and allow for action potential. We think that binding on D1 and D5 stimulate more production of that second messenger or that substance. D2, 3, and 4, we think stimulate the decrease in production of second messenger. So they kind of do the opposite of what D1 and D5 do. Now, some drugs that interact with these receptors, first of all, thorazine, which is the brand name of chlorpromazine. This blocks D2 receptors. So this is given commonly to schizophrenia patients. This is used to try to alleviate hallucinations. Also, apomorphine, this also interacts with the D2 receptor. In low doses, it stimulates the presynaptic receptors or the sending receptors and inhibits production and release of dopamine. So if it is inhibiting dopamine release, it is an antagonist. So it is going to reduce the effectiveness of dopamine. In high doses, it can actually stimulate postsynaptic receptors and become an agonist. So this drug is interesting. In low doses, it can be more antagonistic. In high doses, it can actually be agonistic. We can further discuss agonists for dopamine. So these are, again, going to be drugs that increase the dopamine availability. In particular, they actually prevent reuptake of dopamine from the synaptic cleft. So it allows dopamine to hang out in the synapse. Amphetamines and methamphetamines allow for dopamine to remain in the synaptic cleft by preventing reuptake. In particular, they actually do something quite interesting. They both cause transporters to run in reverse. So instead of transporters removing dopamine from the cleft, they actually run in reverse and propel dopamine and norepinephrine back into the cleft. So again, this is going to increase dopamine availability. Also, cocaine and Ritalin also prevent reuptake of dopamine, allowing it to remain in the synapse. So increase the availability of dopamine. And last is Deprenyl. This does something a little bit different. So this prevents dopamine destruction. So dopamine isn't allowed to be broken down. So again, this is going to allow for more dopamine availability. This is also a drug that's used to treat Parkinson's patients. Now we look at another catecholamine, norepinephrine. So if we look here on the right-hand side of the screen, we're looking at, we just discussed dopamine. So norepinephrine is the next one in the stair-step process. So norepinephrine is synthesized from dopamine. So it goes from dopamine into norepinephrine, and it is done so by dopamine B hydroxylase. So dopamine is converted into norepinephrine by dopamine hydroxylase. Norepinephrine is found in both your central nervous system and your peripheral nervous system. Cell bodies that are involved in the norepinephrine system are located in the pons and in the medulla of the brain and in one area of the thalamus. So activation of these neurons, we think, is related to the increase in vigilance or just attentiveness to what is going on around you. Blocking any part of synthesis of dopamine or norepinephrine will reduce production of norepinephrine. So if we look at the drug AMPT, this prevents tyrosine conversion to L-DOPA. So that is the very beginning of the stair-step process here. So AMPT prevents tyrosine from becoming L-DOPA. Well, if that process is stopped, then you can't then get dopamine and then get norepinephrine. So this is going to block production of norepinephrine. Fuchsic acid is another one that inhibits dopamine being converted into norepinephrine because it inhibits the enzyme that does that. So it inhibits dopamine B hydroxylase. So this is going to block formation of norepinephrine because dopamine can't be converted into it because the enzyme dopamine hydroxylase is being blocked. There are 4 known receptors for norepinephrine, A1 and A2, and B1 and B2. These receptors are sensitive to both norepinephrine and epinephrine. All are metabotropic, meaning that there's not any direct binding that will automatically yield ion channel opening. So if binding occurs with norepinephrine, then it's going to produce that substance that's going to open ion channels rather than opening ion channels directly. So this system overall is going to take a bit more time. It's a little bit slower than that direct activation. A couple of drugs of interest. Idaxazan, this one blocks A2 autoreceptors. So this is given to schizophrenia patients and Alzheimer's patients. Yohimbine blocks autoreceptors. And this one's usually typically used in veterinary medicine, and it's used to counteract sedation. So after surgery has been performed, for example, they want to bring the animal out of sedation. Yohimbine is commonly used. This is going to allow for the increase in heart rate and blood pressure. So if you know anything about surgery, the drugs given typically drop heart rate and blood pressure down a bit. Coming out of sedation, you want to increase those things back up. This drug will do that. Another one is clonidine or catapress, and this one decreases activity to reduce heart rate and blood pressure. So this is a commonly used drug. This is again going to affect perception of norepinephrine. ADHD, drug withdrawal, and menopausal flushing are also related to, are different things that catapress may be given for. Now here we look at the deactivation of norepinephrine. So once it is in the synaptic cleft, it is deactivated from MAO-A or monoamine oxidase A. This is the substance that will deactivate norepinephrine and help it be removed from the synapse. So if we want to increase norepinephrine availability, we might give an MAO-A inhibitor or something that blocks MAO-A. A common drug used is moclambinide. This, again, is a form of MAO inhibitor. You may have heard that term before. These in the past have been used to treat depression. However, more recently, we've kind of moved away from MAO inhibitors because they tend to have some undesirable side effects. So more recently, they've been kind of moving away from MAO inhibitors and moving more toward things that inhibit the reuptake of dopamine, norepinephrine, and also what is called selective serotonin, which we will discuss here in a few minutes. And now here we are, serotonin. Serotonin is another neurotransmitter. It is a monoamine. It is involved in mood regulation, eating control, sleep arousal, pain regulation, and in dreaming. Cell bodies involved in the serotonin system are found in clusters, about 9 clusters to be exact, in the brain. Most are located in the midbrain, pons, and in the medulla. These cell bodies will project out. So they have axons that project out into your cerebral cortex. The precursor to serotonin is called tryptophan. Here we will look on the right-hand side of the screen, much like we did for dopamine, norepinephrine, and epinephrine. Here we'll look at the right-hand side of the screen, and we can see that tryptophan, right here, is the precursor to serotonin. So tryptophan hydroxylase produces 5-HTP, which is right here, from tryptophan. So here is tryptophan hydroxylase. It converts tryptophan into 5-HTP. And then after that, 5-HTP decarboxylase converts 5-HTP into serotonin. So kind of another stair-step process. PCPA will block tryptophan hydroxylase. So PCPA is a drug. If given, it blocks tryptophan hydroxylase. So it blocks this right here. So it blocks what converts tryptophan into 5-HTP. So if that happens, obviously serotonin production is not going to happen either. We know of at least 9 different receptors for serotonin. Most are metabotropic, meaning that they are not direct in that binding with serotonin doesn't automatically open those ion channels. There's an additional step there. So most of them, again, are metabotropic, except for one, which is called 5-HT3. This one is inotropic. 5-HT3 will inhibit postsynaptic potentials by controlling the chloride channel. So if you remember salty banana, the chloride channel, this 5-HT3 inhibits the postsynaptic potential, so the receiving cell. So that potential there will be inhibited because the chloride channel is being affected. So the exchange of ions in and out doesn't work such that action potential is possible. There is a drug called ondansetron, which interacts with 5-HT3. Specifically, it's an antagonist, meaning that it reduces activity on that particular receptor site, so 5-HT3. This particular drug is used during chemotherapy and in radiation to reduce side effects of the chemo and radiation. Another drug that interacts with 5-HT with one of the receptor sites for serotonin is called Buspar. It's a partial 5-HT1A agonist, so it's going to increase activity in this particular receptor. This is used to treat anxiety and depression. So what this is going to do is allow more binding to occur in this receptor site. So more serotonin uptake or more serotonin binding on this particular receptor. Again, this is meant to help treat anxiety and depression. Also, more of a street drug, LSD. It is a direct agonist for 5-HT2A, so it is going to increase activity on the receptor site 5-HT2A for serotonin in the forebrain. This is going to interact with serotonin transmission, and it can actually lead to visual perception distortions because it's affecting receptor sites more in the forebrain, which is related more for visual perception and things like that, visual imagery. So it's going to affect visual perception quite a bit. So drugs that prevent serotonin reuptake are used to treat mental illness. Serotonin, I'm sure you all have at least heard of serotonin and overall have heard that an increase in serotonin might help elevate mood and things like that. So drugs that prevent reuptake or prevent the removal of serotonin from the synapse and then thus increase serotonin availability are used to treat mental illness. Again, they increase the availability of serotonin in the synaptic cleft so that there's actual more serotonin there and it can be received by the postsynaptic cell. It must be given for several weeks, these drugs, to see any kind of therapeutic effects. So this isn't something that you're going to take the drug and immediately see improvement. This is something that requires time to see effect of. Neural adaptations occur during this time. So actual structural changes happen during this time of taking these type of drugs. Examples would be obviously Prozac, that is a very well-known one. This one is used to treat depression, some forms of anxiety, and also obsessive compulsive disorder. Fenfluramine, this drug prevents reuptake of serotonin. This is also formerly known as fen-phen. If you all know anyone who lived through the 90s, or if you lived through the 90s, then you are at least familiar with the term fen-phen. Fen-phen was a diet pill, basically, and it was meant to be an appetite suppressant. And it worked by preventing the reuptake of serotonin, so more serotonin was available in the synapse. And the way that it affected the system was that it suppressed your appetite. Unfortunately, it also caused heart valve damage in many users, and it was directly correlated with a fair number of deaths, actually, because of that heart valve damage, so it was taken off the market. And then another street drug, MDMA, or more so a street drug, also known more commonly as ecstasy. This one causes norepinephrine and 5-HT transporters to run in reverse, so force more of these neurotransmitters back into the synapse. Of course, this is going to yield more of an excitatory response, and this drug can be an hallucinogenic drug. Unfortunately, this drug also selectively damages serotonergic neurons. So the neurons that receive serotonin are damaged by use of ecstasy. This results in a lot of cognitive deficits over repeated usage over time. So our next monoamine is called histamine. Histamine is highly involved in your wakefulness and sleep states. Cell bodies that are in the histamine system are only found in the posterior thalamus. The cell bodies in this system then project to your cerebral cortex and your brainstem as well, which makes sense because the system is highly related to your sleep states and your wakefulness. So it makes sense that these cell bodies would project into your brainstem. Drugs that block receptors of antihistamines, block receptors of histamine are called antihistamines. You're probably very familiar with that term. If you have ever taken a Benadryl, you've taken an antihistamine. And if you think about one of the side effects of an antihistamine, you might remember feeling very drowsy. These drugs can cause drowsiness. Some folks are more affected by these drugs than others. Personally, I cannot take Benadryl. I will be asleep for 2 days if I take Benadryl. So I'm highly affected by an antihistamine. So the precursor to the neurotransmitter histamine is called histidine. Histidine is synthesized by histidine decarboxylase. Histidine decarboxylase synthesizes histidine into the neurotransmitter histamine. Now, your central nervous system contains 4 different receptors for histamine. Interestingly, drugs don't cross the blood-brain barrier, so they have no effect on the brain. So more specifically, they don't make you drowsy. However, a fair number of them do contain diphenhydramine, which Benadryl is a good example again. Benadryl contains diphenhydramine. Diphenhydramine does cross the blood-brain barrier, so it does go into your brain and will cause or can cause drowsiness. Now we move on to peptides. Peptides are all precursor molecules, so they are all the beginnings of something else. They're broken down into smaller neurotransmitter molecules by enzymes. And then they're released in conjunction with another classical neurotransmitter. So they're released alongside another neurotransmitter, like the ones that we've discussed thus far. They regulate sensitivity of the pre- and postsynaptic receptors to the neurotransmitter. So how sensitive the sending and the receiving cells are, or those receptor sites are, to the neurotransmitter. There is no reuptake mechanism or for peptides. So a peptide cannot be taken back in by the sending cell. It has to be removed from the synaptic cleft by enzymatic destruction. So the peptide has to actually be destroyed by an enzyme rather than being taken back in by the sending cell through reuptake. Some that would be included as peptides would be endogenous opioids. Now, endogenous opioids are just a class of peptides that are secreted by the brain that act as opiates, okay? Opiate receptors were discovered first in the 1970s. There are at least 3 types. We call them mu, delta, and kappa. Different neural systems are activated when receptors are stimulated by a peptide. So, for example, some of the results can be analgesia, which overall means numbness. So it can produce numbness. It can inhibit what we call species-typical defense responses. So we as humans have these, but also non-humans have these types of behaviors as well. And they're called species typical because most members of the species perform these typical behaviors. These could be looked at as more fleeing responses, fighting responses, hiding responses. Neurons involved in the reinforcement of drug use. So drug reinforcement that later leads to drug abuse. That is something else that's affected when these receptor sites for peptides are stimulated. Only 2 drug types that affect neural communication through opioids are known. So direct agonists, these would include heroin, methadone, and oxycodone. Then there are also antagonists, so reduce the effectiveness or reduce the availability of peptides. One would be Narcan. This is the drug used to reverse overdoses. So if you've had too much of a drug, this is the one that they're going to give you to try to counteract that effect, or reverse opiate intoxication. So this brings us to our last grouping of neurotransmitter types that we're going to talk about in lecture. These are lipids. So these are more kind of fatty substances. These are substances that are derived from lipids and can be transmitted within or between cells. The best known are the groupings of endocannabinoids. So more particularly, I'm going to talk about THC and the physiological effects that can be felt from THC. THC has become quite popular in more recent history. There's a lot of research being done on whether or not it is truly effective and what are long-term effects and side effects that can be as a result of THC use. The effects of THC and the immediacy are numbness and sedation effects, and that kind of leads it to be used to help combat the effects of cancer treatment. It has also been reported that people who are asthmatic can get good results by using THC. This can also be used to decrease the pressure in the eye from glaucoma. Also can be used to treat motor disorders. The research on this is still being done, so we still don't know fully exactly how effective this is, although some people really think that it is. There are some effects that aren't so great of THC to be aware of. It has been found to affect memory, affect concentration, affect your visual and your auditory perception, and can also distort time. Synthetic versions of THC would include things like spice or K2. These are commonly more available as kind of like a street drug. Now, there are two types of cannabinoid receptors that we know of, CB1 and CB2. Both are metabotropic, meaning that activation of these receptor sites is going to take a little bit longer because when the cannabinoid receptor is activated or binding has occurred on that receptor site, it doesn't automatically open those ion channels. It has to produce the substance that will then open the ion channel. So a little bit slower acting, but again, both CB1 and CB2 are metabotropic. THC stimulates CB1 receptors in your peripheral nervous system. So this is what is going to produce that numbness effect. Tylenol or acetaminophen works similarly in that it affects those receptors from CB1 and it doesn't cross the blood-brain barrier. CB1 can be blocked by a drug called rimonabant. This is an inverse agonist. So this drug can be used as an appetite suppressant. All right, folks, that is all of the neurotransmitters that we're going to talk about. Thank you so much for making it to the end of this lecture. Again, go through this chapter carefully, but please don't have an expectation that you're going to know every term and everything about all of this. It is a lot of material. Please focus on the main groupings of neurotransmitters, what neurotransmitters are, how they can be broken down and removed from the synapse, and just overall have a general idea of the importance of neurotransmitters, the different types, and what they do for us. That's all for now, guys. Have a great day.